Right, this is question 1 of paper 2. Athletes carry out aerobic training in order to improve the efficiency which their cardiovascular system delivers oxygen to their muscles, and the body responds by producing more blood cells. That's quite important, more blood cells. Describe only one adaptation of red blood cells. So there's all sorts of things you can go at here. If I just draw a red blood cell, you could say that they're concave on both sides, or biconcave. That's when it dips in there. We don't particularly need to explain here, it just says describe. We could say that they don't have a nucleus, that they are quite thin, flexible, or indeed they contain hemoglobin. So all of those will do for that 1 mark. This is just a kind of welcome to the question sort of mark, you know, nice easy suggestion. So biconcave shape, that'll get you a mark. But then you could also say no nucleus, thin, flexible, has hemoglobin. Explain how training at altitude can help improve an athlete's performance when subsequently competing at sea level. When you go up to altitude, there's less oxygen there, and so your body adapts to this lack of oxygen by producing more blood cells. So that's what's going on. And if you've got more red blood cells, that means more oxygen can be delivered. So it tells you more blood cells, so you're not getting a mark for that. So more O2 can be delivered to the muscles. The physiology of delivering oxygen to muscles is linked to the removal of CO2, which is mainly transported in the form of hydrogen carbonate ions. Explain how CO2, carbon dioxide, becomes hydrogen carbonate. So in the muscles you are producing a CO2, that's going to go into the red blood cell, and then inside there's going to be some chemical reactions where it's going to become hydrogen carbonate. So what's going to happen is it's going to combine with water. So it's going to diffuse in, combine with water. That will then make carbonic acid, H2CO3. No, H2CO3, that's right. Okay. And then that's going to dissociate into 1H, 2H's, CO3, and that'll be H plus and CO3 minus. And that is the ion that we are talking about, the hydrogen carbonate, or sometimes called bicarbonate ion. So we just need to give those steps. Remember, we've got 3 marks here. So CO2 diffuses into the red blood cell, or you could say follows a gradient of CO2. CO2 combines with water to form carbonic acid. So H2CO3. And we could actually say here, there is a mark for saying the enzyme that catalyzes that. So catalyzed by carbonic anhydrase. And then we can talk about this dissociation. So H2CO3 dissociates into hydrogen ions and hydrogen carbonate ions, into H plus ions and HCO3 minus ions. And now we've answered the question, because we've told how CO2 becomes a hydrogen carbonate ion. Describe how the production of these hydrogen carbonate ions account for the Bohr effect. A bit of a tricky one, this. It relies on this hydrogen. So this hydrogen here, that does something, that answers this question. And what it does is we've got some hemoglobin, and it's got oxygen attached to it. So we're carrying the oxygen on hemoglobin. But what we want to do is get that oxygen off and deliver it to the muscles. And actually, what hydrogen ions do is they will attach to this hemoglobin, and the oxygen will become unattached, or will become dissociated. So we'll get that HAB, and then we'll get the separate oxygens, 4 of them. So that's the point that we need to get over. So H plus ions combine with the oxy hemoglobin to form HAB or hemoglobinic acid, or hemoglobinic acid. And that the key thing is that displaces the oxygen. So this displaces the oxygen, or you could say lowers the affinity of hemoglobin for oxygen, same thing. And that's your 2 marks. A blank page. Then we have got a little bit of kit called the Pulse Oximeter, and that is sending two wavelengths of light, red and infrared, through the finger and presumably through the blood, and it gets sensed. And we have got a graph here. Let's just make sure that's on screen. There we are. We've got a graph, and there's kind of, we need to get this the right way around. So we've got that line there, and then the solid line. So 2 different, and these are for hemoglobin and oxyhemoglobin. Deoxygenated blood absorbs more red light but transmits more infrared light, whilst oxygenated blood absorbs more infrared, but so they do the opposite things to each other. Using the information from the description of right suitable level for the vertical axis. So what is this measuring? So what is this measuring? And the key words there are absorbs. So here we want, absorbs, or light absorption, or something like that. So absorbance, or light absorbed, you might say. We can't mention a particular red or infrared because both are on here, so it wouldn't be true for the other one then. So just a general absorbance. Complete the table below to show the readings which the sensors would give. So you need to find the right line and the right light. So red. So for red we've got here, so that is red, and reading from the red sensor and the percentage saturation of hemoglobin 100%, so that is this one here, and so we're reading this point here, and up to 0.2 it's 10 squares, so each square is 0.02, so that is going to be 0.02 there, because each square is 0.02. And then on the right we need to choose the infrared sensor, so we're now across here, and we need to choose the right line, the 0 line. There's the 0 line, it's the one I've coloured in yellow or highlighted, and so we need to read off this point here, and it's 3 squares above 0.2, so that is 0.26. So it's just, can you read a graph? You get 1 mark for doing both of those. Then part 3, explain why an oximeter would be useful for monitoring the condition of a patient suffering from chronic lung disease. Well, we don't care what chronic lung disease is really, but you can hopefully guess what we're trying to achieve here, and it's the level of oxygen in the blood. So 3 lines, but you only really need to say this is useful to check the oxygen level in the blood. So to check the oxygen level of the blood, or blood in the body. Part D, a double circulatory system is found in mammals, whereas a single in fish. Describe a difference, that'll be 1 mark, and explain why a double is more efficient. That'll be the 2nd mark. So the difference between double and a single, hopefully these definitions are in your revision and in your notes. So a double, the blood travels through the heart twice in one circulation. That's your definition. You could give the single, where the one circuit includes both the lungs and the muscles, possibly. So I'll talk about a double. So blood travels through the heart twice in one circuit. So there's kind of 2 separate circuits, as it were, in one complete circulation. So, or you could say 2 circuits in one complete circulation. You could talk about, single, only one circuit. Heart, lungs, muscle, heart, and one circuit. And the advantages of a double. So advantages, it's really so that we can have as much pressure as possible going to the systemic organs, so your brain, your muscles, and so on, things that are very far away or are against gravity, and whereas the lungs can operate at a lower pressure, so we don't damage the delicate tissues of the alveoli and so on. So advantages: allows the oxygenated blood to be delivered to, I'm going to put systemic, it doesn't say this in the mark scheme, but to remind you, systemic organs under high pressure. And it's key to say high pressure. You could attack this the other way. You could say, kind of, put it on its head, and the disadvantage of a single system where you're losing pressure after the lungs and before the muscles, but it says why the double is more efficient, so I'd talk about that. And the last part of this monster question. The drawing below shows a human heart with its associated main blood vessels. 2 defects are shown. There's actually 3 there. I will admit I only spotted 2 the first time round, but there's a cheeky little third one. The instructions, it says use 2 arrows labelled A and B to show the position of the 2 defects. So don't just write A and B, but 2 arrows. So the first one, quite a major one, there's a hole in this heart. There should be tissue there in the between the left and the right ventricles. They should be separate. We just talked about double circulation, and this one actually there's a problem with our double circulation, so it's not separate. So we'll have that as A, and we've got a sneaky little connection here between these 2, the pulmonary artery and the aorta. We seem to have a connection between them, so again that is connecting up the 2 sides, so we'll call that B. There is another one, which is this valve here. It's not kind of in 3 parts. It should have 3 cusps to it. I didn't spot that the first time. So there's 3 to go anyway. So suggest the consequences, and you are thinking about problems with this kind of blood mixing, really. So blood oxygenated. So oxygenated blood on the left hand side would possibly go into the right hand side and be sent to the lungs, or deoxygenated blood might go the other way. So oxygenated and deoxygenated blood would mix in where we've labelled A. So that's a problem. And in B we've got a similar sort of a problem where we've got pulmonary artery and the aorta. So blood from oxygenated blood might go to the lungs. So for example, so that's just A and B. So problems that you might get oxygenated blood going to the lungs. That's a problem. And another problem might be deoxygenated blood going to body systems like muscles. And it says explain why the oximeter readings would be different from a person without. So what would happen to the oximeter? That would be showing a lower reading. So oximeter will detect less oxygen. And the other answer from the sneaky little third error, with the faulty valve there, you could say it would allow the backflow of blood from the aorta into the left ventricle.